This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWIM. This is TWIM, This Week in Microbiology, episode 176, recorded on May 10th, 2018. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schechter.
Hello there.
You know, last time we recorded was your 90th birthday.
It was.
Happy birthday.
I'm a nonagenarian.
Nonagenarian. Happy birthday.
Thank you very much.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How are you, Michael?
I am well.
You're getting ready to go to Atlanta?
I'm getting ready to go to Atlanta, but before I go to Atlanta, I'm going to go to Clearwater Beach, Florida, where I have the pleasure of chairing the 17th workshop for teaching microbiology and immunology to medical students, a conference that was started by the micro chairs some 30-odd years ago.
Nice.
And so I've been chairing this meeting on behalf of the AAMC group for the past, this will be my third time that I'm chairing this meeting, and it meets every other year. It's sort of like Q, but for medical school.
So what's the key to teaching microbiology to medical students, Michael?
It's the theme for this year's meeting is winnowing the curriculum. What does a graduating medical student need to know about microbiology and immunology? And unlike the other disciplines, and I'll probably get hate mail for this, but anatomy really isn't changing all that much, whereas micro and immuno is evolving at a great clip.
Mm-hmm.
You just have to look at the advances in medications that we're developing for cancers that are specifically targeted to checkpoints in our immune system.
In other words, there's no sense in teaching them anything.
Yeah, this is true.
By the time they graduate, it's not going to be relevant.
Well, yeah. And, the great debate this year is, do we need to be teaching about selective and differential media in an era of MALDI-TOF?
That's a great question, yeah.
Do they really need to know about?
No.
Well, this is good. But unfortunately, the people writing the questions for the national board examination are us. And unfortunately, there are sticks in the mud who think we need to still be teaching medical students wet labs and all sorts of other things. And there's a lot of blood on the floor after some of these discussions, but
sure, yeah.
It makes for a lively activity.
By the way, I am all for having a wet lab.
Uh-huh. But you want to have a MALDI-TOF in that wet lab.
Yeah, I'd like to have a MALDI-TOF, and I'd like for them to appreciate how to do a gram stain, especially from fluids like spinal fluid, and really teaching them the finesse associated with it in concert with drawing that fluid. So they really begin to appreciate the importance of specimens and specimen collection and things along those lines.
Let us know how it went, will you?
I will when we maybe we'll have a little discussion about it at our next meeting when we get together in Atlanta, which will be shortly.
Good.
Gee, Atlanta's in the beginning of June, isn't it?
It's coming quick.
Yeah. Well, we have one more here in studio, and then Atlanta. That's right. All right. We will first tell you another unfortunate passing in microbiology. Last time we told you about Alan Campbell. This time, Stanley Falco has passed away. He's also from Stanford University, as was Alan Campbell. And Elio just noted before we started recording, Charles Yanofsky passed away some time ago. He's also at Stanford. Did any of you know Stan Falco?
I did. I didn't know. I knew him pretty well, in the sense that we'd get together at meetings and have a cup of coffee. That was the extent of it. But one of his first graduate students of his modern era of molecular pathogenesis. It was Ralph Isberg,
Mhm.
who joined my department at Tufts. So indirectly, I got to know Stan quite well. So let me say something about him. He is considered the father of, quote, molecular microbial pathogenesis. By this, what is normally meant is that he started the idea of using genetics, using mutants, to determine what the variance factors are for a certain organism. And he codified that by calling it the molecular Koch postulates. It is, what is it that you have to do to demonstrate that a factor is indeed a variance factor? In the process, he trained innumerable people. The field is rife with Falco alumni. They are all over the place, and they all learn from the master that you can make things rather straightforward and simple in the world by asking the right question. And he was a good guy. He had a wonderfully foul mouth, so it was fun to listen to him talk, and a very friendly, very warm person, I would say.
How about you, Michael? Do you know?
I did. In my early days on the ASM Education Board, Stanley was president of our society at the time, and he asked me to chair the search committee that was looking for a replacement of the Education Board. Fred Fender had timed out, and Stanley was looking for someone as good as Fred or better, and we ended up getting a new head of the Education Board, and that individual ultimately became one of the society's presidents, and that was Cliff, and I'm blanking on Cliff's last name.
Well, I knew Stanley. I met him the first time when I was looking for jobs back in 1981. I interviewed in he was chair of microbiology at Stanford, and I interviewed, and he offered me a position, and I ended up coming here to Columbia.
As opposed to California.
Yeah, I didn't really want to go. Well, the reason I didn't go was because there was really no virology at Stanford at the time. And here there was at Columbia, and I made the right decision because I had a lot of good collaborations here. However, now I would like to be in California. Now, at this point in my career, it would be nice, but of course, it's too late. And the funny thing is, many years later, I think 15 years later, I visited the same department at Stanford, and there was someone there who was a faculty member, and he was a student when I visited, and he remembered me saying, I don't want to come here. I don't like palm trees. Anyway, so Stanley was a lot of fun. Yeah, he was. And then after that, I knew him. I saw him at meetings, and he was just a wonderful speaker.
Yeah, really a major figure, no question.
And I want to refer to a blog post he wrote for Small Things Considered. We'll put a link in the show notes. Everyone should read it. It's called Fecal Transplants in the Good Old Days.
And I'd like the ASM president and former chair of the Education Board was Cliff Houston. I don't know how I could forget Houston's. Houston, but I guess I'm blanking on cities today. But the other thing we should mention about Dr. Falco is that before he became Dr. Falco, he was a medical technologist.
That's right.
Of the ASCP. So he really did bench level microbiology for a long period of time. And I got to know him pretty well at a Gordon conference. Was the year that ASM had a Gordon conference in honor of education. And I was one of the speakers at that meeting. And I remember having breakfast with Stanley, and we were talking about or lamenting about the cost of sequencing and finding virulence mutants. And he shared with me his quick and dirty way of finding a strain of bacteria that would have a pathogenicity island. And this was at the time pathogenicity islands were emerging and people were beginning to understand the importance of them. And he said, I always knew it was about the IS elements. I always knew. And he would just scan for these insertions.
Besides being trained in clinical microbiology, he was a wonderful geneticist.
Oh, absolutely.
He really is responsible for us understanding plasmids the way we do in a major way. So
and he and Alan Campbell, and if you have not yet seen Elio's tribute to Alan Campbell, I encourage you to go over to Ilio's blog and take a look at the fine piece that Ilio wrote about Alan Campbell.
Thank you.
And
Thank
you get to learn of the wonder of the U.S. draft and all the wonderful things that it did. That was a great piece, Ilio. I really enjoyed
very
reading.
much. Appreciate
I
that.
really enjoyed reading it, and I learned things I had not known about either you or Dr. Campbell.
I heard a quote from Stan Falco last week. He said, this is pretty funny, and it's also good. The goal of a bacterium is to become bacteria.
Yeah.
Lovely.
Isn't that typical of Stanley,
I think that's
right?
the goal of
That
humans,
sounds like
too.
him.
They become humans? I don't know. Not everyone, not everyone.
Yeah.
I want to tell you about the upcoming meeting called ASM Microbe, the annual meeting of the American Society for Microbiology. Anyway, this year it's in Atlanta, Georgia, and ASM has a special opportunity for our podcast listeners. Get $50 off registration for Microbe 2018, which is June 7th through 11th in Atlanta, using the promo code ASMPOD. ASM Microbe 2018 connects scientists with their science and showcases the best microbial scientists in the world. Delve into your scientific niche in 8 different tracks. Visit asm.org/microbe. That's asm.org/microbe and use the promo code ASMPOD. It's all one word for $50 off registration. All right. Now for our science, we have a snippet from Elio.
I got a snippet. Man, do I have a snippet. Listen to this title. Converting Escherichia coli into an archaebacterium with a hybrid heterochiral membrane. And it's by 10 authors. So I'll just mention the 2 top and the 2 bottom ones. Antonella Caforio, Melvin Ciliacaus, and at the other end, Arnold Driesen and John van der Oost, all from Holland, many from Groningen, actually. Anyhow, get this title, Converting Escherichia coli into an Archaebacterium. Right there's a problem. Why Archaebacterium and not Archaeon? Well, that's for the authors to decide. They don't go into that. But the idea is that if you were to look at the differences between archaea and bacteria, many stand out, but high on the list is difference in the lipids. Okay, so bacteria have regular phospholipids the way we know them. They have a phosphatidylglycerol to which the lipids are attached at the 3-carbon, just like it's also true for eukaryotes. Interesting. Whereas archaea have the lipids, the fatty acids rather, or isoprenoids, stuck to a 1-carbon on the glycerol, and it's by an ether link, whereas in bacteria and in eukarya, it's by an ester link. So these are very fundamental differences. In addition to which, and this is sort of an aside, there are a few archaea which have another puzzling thing. Instead of having a lipid bilayer, they have a lipid monolayer. It's like it's as if the tails of the fatty acids were linked together. All of this put together is said to be because so many archaea are extremophiles. That is, they live at very high pH, very low pH, very high temperature, high salinity, etc. They're not all extremophiles. Some live in the rumen of a cow, but many are. And so the idea is that this great distinction in lipids between bacteria and archaea has something to do with survival. Okay. Now, given that, you would never think that you could clone the genes for making these different different lipids into the ones from archaea into a bacterium, and therefore converting, as the title says, Escherichia coli into an archaebacterium with a hybrid heterochiral membrane. So how do they do it? Well, they take a bunch of genes from both archaea and bacteria because the paths overlap some, and put them into E. coli. They put it under a nice high-level expression system, which consists of a LAC promoter, which is tunable. You can just add IPTG or whatever, and you turn the thing on, and you put it in an area of the chromosome of E. coli, which is highly expressed, namely OREC, the origin of replication. Happens to be very convenient for cloning purposes. So now, first of all, let me back off a second. Lipids always get, not always, often get a low exposure in the scheme of things. If you don't need them, you forget about them. And so
that's right.
So I believe that they need their proper respect, and they deserve a better place in the biological limelight. So I welcome the emphasis on this. But to believe that you can do this, they convert an E. coli into almost an archaeum with regard to the lipids, is really a fantastic stretch. I mean, it shakes your faith in something. Is nothing sacred? So here we are. So anyhow, they do this, and they figure out that if they do it the right way and play around with it, and it's complicated, I won't go into that, it's being a snippet, but they get up to 30% of the lipids of this engineered strain to be archaean lipids. Okay, that is significant. It turns out that the enzymes involved in making putting fatty acids on a 3 position and in 1 position of phosphatidylglycerol are unrelated. So it's not a question of borrowing a little bit from a cognate enzyme. These are very different enzymes. They also tamper with the polar head group to make everything kosher. And so they end up with this incredible cells. It's an E. coli, which has 30% lipids. It shouldn't. So what's it like to be an E. coli with this load of lipids? Well, it depends how much you induce it, because you can induce it to various levels by adding different concentrations of the inducer. And if you do it at moderate amounts, which still have about a lot of lipid, this 30% comes from that, the bacteria are said to grow at about the same rate as the parent strain. Let me take a break here. I have a real bone to pick here, because they show growth on a linear plot. Well, I don't know why they do that, because growth is exponential. And so when you look at the plot, you can't tell, you can't compare one with another. But that's what they say. So let's let it go.
And I don't know how they can get an OD of 5.5.
Oh my God, that's another thing. Everything is done at enormously high concentrations of bacteria where growth is not exponential. So I have no idea whether, in fact, this statement is correct or not, but let's just live with it and go with it. Okay, so what else do they know about it? They know that if they overexpress these genes, things are not so nice. The cells become elongated, they become funny looking, and they apparently grow more slowly as well. So you can't just keep pumping the cells full of lipids that they don't belong to them. But you can do it reasonably and get a system which is amazing. It is nothing short of stunning. A bacterium which pretends to be an archaeon in part. So what else do they know? They know that these cells are indeed more resistant to environmental challenges. For instance, they withstand exposure to a higher temperature, 58 degrees, whereas the control strains don't survive 50 degrees. I'm talking about centigrade. They're also better able to survive freezing and thawing, as well as exposure to butanol. I would say that this kind of work is going to be followed up. It needs more work, but right now the indications are that these engineered organisms are in fact have greater resistance and therefore maybe higher fitness. And as they say, this has possible biotechnological applications, because if you can make the cells where you've engineered something in, besides
50 degrees. I mean, just look at the temperature profile. You can transform things at higher temperatures and make it just so much more efficient.
Right.
And
exactly.
that swapping out the ether-linked lipids for the ester-linked lipids, if that gives you a delta of at least 10 degrees C in temperature, it can make efficiencies of fermentations as you try to mass produce useful products, and it's really fascinating. The other thing I found interesting is how they started it with introducing the audience to LUCA. LUCA is the last universal common ancestor. And, just thinking about LUCA, the last common universal ancestor, and the fact that the bacteria and the eukarya have these ester-linked lipids, whereas the archaea are using these ether-linked lipids, and they actually sort of tease you and tell you that in ancient times, when the common ancestor was around, there was some thought that the way cells may not even have had membranes. They may have been using such, heretical things like iron to contain cellular processes. So with these iron sulfide
spend a fair
structures,
amount of time. They introduced the whole paper by going through this, and I mean it's perfectly legitimate, and they're totally entitled to do it in view of their amazing results. But I would say that all this is still, let's say,
theoretical.
very much the realm of speculation.
Yeah,
and speculation is fun, but it's best done over a beer.
Well, the Netherlands make fine fermented products.
That's
so
true.
You can have many beers in Holland, and think about these things.
Maybe that's how this comes about. Anyhow, this is an amazing paper, and it does talk, if anything, about the incredible plasticity of bugs like E. coli. We discussed, didn't we, a paper where somebody cloned a gene for a single protein from a eukaryote named caveolin into E. coli. You remember when this was?
Yeah, we talked about it. Yeah.
So this was totally incredible, because cloning a single gene for a protein, which is essentially a membrane protein, makes the E. coli replete with membrane vesicles. I mean, it is nothing but wall-to-wall membrane vesicles. And this is a single protein, a single gene that needs to be cloned. So we're not done with amazing stories like this. And I would say that tuning in on the miracles of the microbial world is well worth doing.
Tune in.
So,
Elio, getting back to the title, I think they call it an archaea bacterium because it's a hybrid, right? It's got a...
Well, yes, except that when you do that, you should explain it. I mean, I think they could have done well. I don't think it appears, the word Archaebacterium, I'm not so sure it appears again in the paper.
Maybe anywhere else in the text.
Yeah, that's true.
Yeah.
Right. So, I think it would be good if they... it's.
But that's the old name for archaea, right? They used to call them archaebacteria.
It is. But archaea. I mean, the people like Norman Pace, who are such a champion for the concept of the three realms of life, the three domains of life, would shudder at the term, because to him it means that there is something in between. Well, obviously, this paper says you can make something in between, but it's artificial, and I think I would be careful with the term.
I mean, I think the idea of converting is a little bit of a stretch, right? Because they provided it with a very different membrane, and that's about it.
But the ribosome is still a bacterial ribosome. It's not a eukaryote. It's not a.
Yeah, that's right.
All the rest is E. coli, and they didn't do the archaeon ribosome or any of those other things.
Yeah.
But that may be their ultimate goal, is to see whether or not they can really goose the system to get it to go like an archaeon. But again, it gives the initial reader of microbiology some insight as to why methanogens would benefit from having these ester-linked lipids, or ether-linked lipids, because they make them more resistant to solvents.
Well, but they do. Methanogens are archaea.
Yeah, that's what I meant. That's why they would. That's why they're in the cow's rumen, is because of the solvents that are being produced from the fermentation products that are coming off.
Yeah, but that's true also for high temperature. I mean, many
That is
archaea are
true.
hyperthermophiles, and it goes for all the extremophile mode of life of archaea. They all may benefit from having their own kind of lipids. It just makes sense.
Yeah. All right. Thank you, Elio.
My pleasure. This was fun.
I have a paper for you from Cell. It is called Microbial Genetic Composition Tunes Host Longevity. And this is all about trying to live longer, at least for worms.
If you're a worm.
If you're a worm.
If you're a worm.
I think humans not do pretty well already, right?
Yeah. We make it to
good years.
90. We make it to at least 90. Our goal is to follow Elio.
So the first author here is Bing Han, and the last author is Meng Wang, and this is from Baylor College of Medicine and the University of Texas, both in Houston. And the idea here is that there is some evidence that our gut microorganisms are linked to the aging process in some way. And the idea is that the microbiome makes metabolites that are important, but also can deal with exogenous chemicals that may be harmful. And we know, for example, that there are changes in the microbiome as you age, and there are diet-driven changes in the microbiome that help your health as you get older. But this is all mostly observational and correlative. And they wanted here in this paper to try and get some direct evidence for the role of microbiome in longevity.
And these experiments are hard to do. And as they introduce us to this concept, there's one line that you got to keep in the back of your head as Vincent begins to reveal their data. And you have to keep in mind or bear in mind that the mammalian gut microbiota is of high complexity, and there are profound technical challenges in isolating specific pro-longevity microbial variations just by getting the sample out of a mammal in the nick of time.
And ain't that the truth?
Yeah.
I totally agree with you, and it has to be said over and over and over again, because the microbiome is part of the daily headlines of a newspaper these days, and the claims that are made are, even if they are couched in tentative terms, to me they sound like hype.
Yeah.
Yes.
Yeah.
And we have to be very careful, but this is a very careful and well-conceived, laid out study.
And so in part it's because they start with a very simple model organism, Caenorhabditis elegans, the wonderful soil nematode that has been a mainstay in many labs because it has a defined number of cells. We know where each cell comes from. And for this paper, it's genetically manipulable. As you'll see, there are tons of mutants available in all sorts of phenotypes. And it's easy to grow in the lab. And it has one microbe in its gut microbiome, E. coli. And you feed C. elegans, E. coli, and that is what ends up in the intestinal lumen, a gut microbiome of N equals 1.
It's the ideal model system.
Right. And so what they do here is they say, we have a E. coli, a K12 strain. We have a library where each isolate has a single gene knockout. So these are genes that you can knock out, and the E. coli will still grow. And there are 3,983 such E. coli mutants. And they simply ask, if we feed each of these to C. elegans from when it's born to when it becomes an adult, what happens? So they go through 3,983 E. coli mutants, and they grow C. elegans on each one of these, and they ask, are there any that prolong the life of C. elegans? And if you're wondering how long C. elegans lives, it's about 25, 28 days or so, looks like from these graphs. And then you can look at a few days, a 5-6 days increase, and that's what they do. So they go through this whole library. They identify 68 mutants, which, again, when fed to C. elegans, they prolong the lifespan. and they validate these. They get it down to 35 mutants. They reintroduce those to make sure that they still work. And in the end, they have 21 mutants that make C. elegans live longer.
And you should point out that these are deletion mutants.
They are.
So they're taking something away from the bacterium that then allows the host to live longer.
Correct. That's right. All right. So you can provide these bacteria to C. elegans when they're already adults.
Because they're eating them.
So in other words, if someday this works in people, you and I, Michael, could take whatever it is they're going to develop and it might help us at our old age.
That's right.
Although I'm much older than you. You know, you're a young guy.
Not by much.
So 21 mutants, 20, and they prolong the life of bacteria. And these genes that are deleted are in all kinds of classes. They have a table here. They're transcription, translation, metabolism, respiration, membrane transport, proteases, chaperones, and other things. So they're all over the place, not just one thing.
And then the famous unknown genes.
Or
the other.
All right. So now they do a series of experiments which leverage these 20-some E. coli mutants and also C. elegans mutants, because there are lots of those as well. All right. And
by the way, the numbers are pretty impressive because some of those mutants really extend the lifespan by a factor of 40%.
Yeah, it's good. Yeah, if you're a worm, you could be happy about that, right?
Yeah.
All right. So first of all, they ask, would these work in a sick worm? Would these help sick worms not be sick? And they have two models where one mutant of C. elegans, you get germline tumors and death. And in fact, if you introduce 16 of these bacterial mutants into that mutant C. elegans, it increases their survival. So if the worm is going to develop a tumor, these E. coli will help it live longer. And the other model, which is interesting, they have a model for Alzheimer's disease in C. elegans. They put in an altered beta amyloid protein encoding gene, and these C. elegans die early. They have trouble moving around, and if they put the bacterial mutants into them, 14 of the 29 prolong the lifespan of these transgenic C. elegans strains. So, in other words, not only do these genes, removing these genes from E. coli, make the worms live longer, but they can also make them live longer in the background of specific pathologies like tumors and amyloid beta accumulation.
And that's at the germ level of the animal, if you will.
Yeah, it is. All right. So now next, what's going on here? It's already been studied in many labs what makes worms live longer, and there are a number of mechanisms. Two of them are the insulin-like growth factor pathway and the target of rapamycin signaling pathway, and caloric restriction. I don't know if everyone knows this, but did you know caloric restriction can prolong lifespan?
It's certainly been demonstrated in mice.
Yeah, it's amazing. You eat less, caloric restriction in laboratory animals prolongs life. It's quite clear. So, in our American culture where we tend to eat a lot, it might be not a bad idea to cut back what we eat.
It's a widely held view.
I just love food. It's hard. Okay, so then they look at some of these pathways. They have mutants which disrupt this insulin-like growth factor pathway, the TOR, and the caloric restriction. And, in fact, they find that some of these bacterial mutants cannot prolong the lifespan of C. elegans on the background of these mutations in the other pathways. In other words, suggesting that they're working through the same pathway, the insulin-like growth factor pathway or the TOR signaling pathway. So that's a very nice genetic approach, right? Because you take out the bacterial gene, the C. elegans gene. If you don't get longevity anymore, extension anymore, then they must be in the same pathway. Pretty cool. Then they look at caloric restriction, and they ask, okay, do these bacterial mutants have any effect on eating or defecating, which are two things that are known to affect longevity in the worm? And they decide that these bacterial mutants don't affect pharyngeal contraction or defecation. So they don't think that these bacterial genes are contributing to those activities. Now, in the course of this study, they find that there are two bacterial mutants in two separate genes which regulate the biosynthesis of cholanic acid, or Ca. And this is a polysaccharide secreted by many enterobacterial species, including E. coli K12 that we have here. And there is a biosynthesis gene cluster that regulates the production of cholanic acid. There's a central transcriptional activator called RCSA, and there's a protease called LON. I'm sure that Michael and Elio remember LON for other reasons.
Oh, that's Matt Gottesman's work, isn't it?
Yes, they got a Gottesman.
Way before that. Way before
that.
It goes back to people in the Pasteur Institute.
So LON is a protease that can repress the biosynthesis of cholanic acid. So their idea is that overproduction of cholanic acid is what is happening in these bacterial mutants. So they first look at cholanic acid levels, and they find that in bacterial mutants lacking the LON protease and another gene that is a positive regulator of that, they have higher cholanic acid secretion. Of course, if you take out the RCSA gene, which is, again, the master regulator of cholanic acid biosynthesis, you suppress the production of cholanic acid. And then, if you put all these mutants together, they complement each other. So you take away the protease, it will suppress the problem caused by the removal of the transcriptional regulator. So in other words, there's a cluster of genes involved in the synthesis of cholanic acid, and they think that this is involved in prolonging life.
So, Vincent, let me ask you, Roy Curtis, if my memory is serving me right, published a paper probably 5-6 years ago on cholanic acid, in which he was showing that it was needed to promote biofilm formation. And one of the things I always associate with biofilm formation is that the microbes are essentially quiescent. They're not metabolizing or doing anything. So do you think that could be, impacting longevity as if it's going to promote E. coli to form a biofilm in the gut of the C. elegans? Or so I was thinking about this, and I'm trying to make the connection of why colonic acid, or are we going to run out and all get colonic acid tablets to begin to take?
I think at the end of this paper, that's what
that's what they're saying.
That's what they're saying. Maybe we should take colonic acid. Yeah.
Yeah.
I think it's not a biofilm because they show in the next set of experiments, basically. You can feed the C. elegans colonic acid, and it prolongs their life.
Yeah.
So you don't need a biofilm. So the bottom line here so far, we have a number of bacterial mutants that give C. elegans a longer life, and some of those genes that are deleted are involved in the synthesis of colonic acid. So there's a link here between colonic acid production and living longer in C. elegans. And they also think that there's a link with mitochondria in these mice. Mice.
In these worms. These are worms.
I know. I'm used to saying mice.
No legs. No legs.
So one of the hallmarks of aging is mitochondrial dysfunction in the body wall muscles of C. elegans. It's a hallmark, and this happens when the mitochondria fragment. And they can take pictures of the muscles of these C. elegans, and you can see the mitochondria fragmenting as they age. And in fact, if you give these C. elegans these colonic acid overproducing bacterial mutants, it attenuates the mitochondrial fragmentation. So that's somehow another link to making the worms live longer. They also show that this works in other nematode species that are 100 million years diverged from C. elegans. So it's not just a C. elegans thing. So that's pretty neat. So the next series of experiments, they get purified colonic acid and they give it into E. coli, and it prolongs their life. It prolongs the life of E. coli. It works with a number of bacteria that you feed to C. elegans. So in giving worms colonic acid also delays the mitochondrial fragmentation. It helps the worms to move better. It helps get rid of tumors. It decreases the beta amyloid problem. And guess what? It even increases the life of fruit flies.
Yeah, that's what was remarkable. And we're talking about wild type Drosophila living as long as it looks like 90 days for a small fraction of the population.
So I have to know what's with mice. Let's feed mice colonic acid, right?
Yeah,
Let's find out what's nice. So colonic acid is, if you'd like to know what it is, it's a repeating unit of glucose, galactose, fucose, and glucuronic acid decorated by pyruvate and acetate. What an interesting molecule. So go ahead.
Go out and buy stocks and people who make colonic acid.
That's right. That's right.
Well, it almost could be peptidoglycan. It's sort of, repeating subunits of simple carbon.
It's not cross-linked, though, is it?
No,
it's not cross-linked.
It's not cross-linked.
But they made monomers of colonic acid, and the monomers don't work to extend the lifespan. They have to be multimers. So, and you can. The colonic acid doesn't affect the bacteria. They did a number of experiments to look at that. And in fact, you can inactivate the bacteria and feed it to C. elegans along with colonic acid, and the colonic acid still works. Now, here's a very interesting experiment. They have mutants of C. elegans that are defective in endocytosis, which is a way of taking up material from the cell exterior. And these mutants cannot have their lives prolonged by giving them colonic acid. Apparently, the worm needs to endocytose colonic acid to have this prolonging of its life. It's really interesting. The next series of experiments, they find a connection between colonic acid and mitochondrial fragmentation. In fact, they have a couple of worm mitochondrial mutants. They have a mutant which is defective at mitochondrial fission, which is the breaking up of mitochondria, right? The dividing. And then they have a mutant defective in mitochondrial fusion. And they want to know, does the colonic acid work in each of these? And what they find is it doesn't work in the fission mutant, the mitochondrial fission mutant. So somehow mitochondrial fission is important in this prolongation of life in worms.
Do they have a clue as to why?
So the next series of experiments, they think that there is an unfolded protein response in the mitochondria. So to just tell you a little bit about that, unfolded protein responses, I'm familiar with it in the endoplasmic reticulum of eukaryotic cells, where if there's some kind of stress on the organism by making different, like viral proteins in the ER, you will have a response to shut down translation because the cell doesn't want to make misfolded proteins. And so apparently there's one in the mitochondria, and they think that the colonic acid is inducing this unfolded protein response. And then the effectors that come in to try and fix that are the ones that prolong the life. They don't really know what they are, but they think that that is the mechanism of prolongation of life. So in other words, and they address this by looking at some very interesting C. elegans mutants that are defective in these unfolded protein responses. And that the effect of bacterial colonic acid on extending the life of the C. elegans requires the mitochondrial unfolded protein signaling pathway. So the idea, Elio, is that the response, the unfolded protein response, the effectors of that response must be somehow involved in prolonging the life. It's kind of counterintuitive that you would need mitochondrial fission to do this, but I think that's because it stimulates this unfolded protein response.
Or you effectively get a new mitochondrion.
You do.
Because when you fizz, you're effectively making a new one.
You make a new one, yeah.
And so you effectively are starting over, and your mitochondria is not effectively senescing or having mistakes and not doing what it was supposed to do.
That could be part of it as well.
So the colonic acid is effectively telling the cell, hey, you better divide because things are bad.
Maybe.
And
part of it.
It may be something simple like that. And it makes also sense why the endocytosis mutant doesn't work, because the colonic acid's not getting in to effectively stimulate the mitochondria.
Well, that could be, or if you don't have the right bacteria that are producing it, right?
Yeah.
Or if the microbiome's changing as you age, maybe you lose the colonic acid producers. I'm sure it's more complicated than that, right? This is one. I mean, these were a handful of bacteria that they focused on, bacterial mutants.
And it's a very, very simple microbiome because it's just E. coli.
Yes, he's right. In fact, they point out that when they look at the genes involved in producing colonic acid, they also see genes involved in chorismate metabolism, which is a precursor for aromatic metabolites. And it's known that deficiencies in two chorismate derivatives, ubiquinone and folate, are why E. coli mutants that have been previously studied are able to prolong the host lifespan of C. elegans. Now, there's a drug called metformin, which interferes with bacterial methionine biosynthesis, is also coupled with chorismate metabolism. And again, that extends life of C. elegans. So they say that chorismate biosynthesis in your microbiome may be another way to promote host longevity in a broader sense, right? Because it wouldn't involve just colonic acid. So
metformin is the one of the most prescribed drugs on planet Earth for folks with type 2 diabetes.
Is that right?
Yeah.
So they say in the discussion, colonic acid may provide an exciting approach for improving human health and longevity. Now, I don't know. I think, as we said earlier, humans can live long. The question is, how healthy you are as you age. So maybe you could address some of those issues with something like colonic acid or something more.
Well, we have a large cohort of folks over 100. We have a large cohort of folks over 90, 80, et cetera. We could effectively measure the colonic acid in those individuals and just ask the simple question, what does the population look like? If it varies all over the place, do a simple correlation experiment may be the first step.
I'll be glad to donate.
You'll donate?
Donate. Now, they end up with a very interesting point here, and that is, of course, mitochondria came from bacteria, and they share metabolic pathways with bacteria. And so it's interesting that the bacterial colonic acid that they study here, that one of the targets at least appears to be mitochondria. So, is that remnant of a previous property? Who knows? It's very interesting.
You're witnessing a family reunion.
Gut bacteria of C. elegans make colonic acid. It induces mitochondrial fragmentation, and it turns on this unfolded protein response. And then that makes some signals that improve host fitness. And that's the big question. What are those signals? Let's find out what they are, and maybe we can then move up the evolutionary ladder here and look at mice and rabbits and then humans, I guess. Anyway, I thought this was interesting because you have the genetics of E. coli and C. elegans combined here to solve this problem. Really neat.
And it's a whole new world of crosstalk. I mean, the microbe, the bacterium, in this case E. coli, is talking to a eukaryotic symbiote, the mitochondrion, which once upon a time was a bacterium.
That's right. Now, Elio, you must have lots of colonic acid.
I'm loaded with it. One molar colonic acid.
All right. Before we wrap it up, just a couple of emails here. The first one comes from Amir. Hey, guys. Hello from sunny Israel. 83 Fahrenheit, perfect beach weather. Thanks a lot for the podcasts. I'm really enjoying them. I'm hearing a lot recently about the E. coli outbreak in romaine lettuce and was wondering, do you know how they trace the source and the specific bacteria? I mean, a person who gets sick suffers from, quote, diarrhea, vomiting, and severe stomach cramps and can lead to kidney failure, end quote. This is a very general symptom of enteric disease from what I know. And if you take a look at the gut microbiome, there's a good chance of finding very similar strains of E. coli in there. So how do they clinically differentiate this strain? How do they trace it back to the specific lettuce growing area? Thanks again for the great work both on TWIM and Small Things Considered.
Thank you.
Well, that's good old-fashioned shoe leather epidemiology. They actually go out and they interview folks who have gotten sick, and this is the CDC and the local public health departments associated with states where individuals have been reported ill, and the CDC will actually call you. My brother got sick once at a wedding, and he and his wife got called by CDC epidemiologists inquiring what they ate, when they ate it, when they got sick. And it's, good old-fashioned shoe leather epidemiology. And then, based on what you report to the CDC, they may elect to ask for a stool sample, depending on whether or not you've had antibiotics. And in this particular case, with E. coli OH157H7, the O stands for the O antigen, which is in the outer membrane of E. coli, and the H is actually the type of flagella protein that this particular E. coli is having. So that license plate is actually a description of the car that hit you or is responsible for the diarrhea that you're getting. And the way they trace it back to the particular farm, and in this case, it's in Yuma, Arizona. And this must be one of those mega factory farms because it seems to have supplied 12 states and a large number of restaurants and stores. And, it's really caused a great deal of concern. I saw a report of it on our local news last night, and that folks are still worrying about it happening here in South Carolina, where this is the height of tourist season. And again, the CDC and our local public health department is very much concerned. But the general guidelines out there to how to protect yourself is, simple hand hygiene. Wash your hands. Don't prepare food or drink for others when you're sick. Keep hot food hot, cold food cold. Cook your meats thoroughly to kill off this nasty variant of E. coli, and avoid raw milk and other unpasteurized dairy products and unpasteurized juices. Fortunately, for most of us in the U.S., we only consume pasteurized milk and pasteurized juices because we have learned our lesson with unpasteurized products. And again, this is one of those types of food poisoning for which you do not need an antimicrobial, because if you do, it can often result in you developing hemolytic uremic syndrome, where the microbe actually dumps more toxin based on your exposure to the antibiotic. And so you don't necessarily want, if you got old antibiotics in your medicine cabinet, you don't want to take them because they may not do the trick.
So, Michael, the difference between O157H7 and the E. coli in your gut is also a lot of other DNA, right?
Oh, yeah. It's 40% different in DNA sequence than garden variety E. coli K12. 40% different in its DNA, yet it's still E. coli.
Do they use that to diagnose at all, or is it just the
what the CDC is ultimately doing today, years past, they would use pulse field gel electrophoresis, which is effectively that molecular fingerprint you see on many of the crime TV shows. But today what they're resorting to is DNA sequencing to identify the culprit. And they're looking for the particular toxin gene for this hemorrhagic toxin that actually came from Shigella,
right,
on a pathogenicity island.
So, Michael, what is the source of the E. coli in the lettuce? Is it contamination?
It's water.
Water.
It's water. It's either water, contaminated water, because it's not potable water that they use to irrigate the fields. As you can well imagine, lettuce is a vegetable that has a very, very high water content. It gives it its crispness. And if you're buying the prepared lettuces, it can also be a function of the wash water. If a product has been contaminated and the fecal material spreads in the wash tank, the E. coli can effectively multiply. And when the temperatures are right, as you're transporting it home, or if it doesn't stay cold in the cooler at the store, and all of these other things can come in to mix it up. And the other thing about E. coli 157.8.7 is for the garden variety E. colis that typically give us traveler's diarrhea, you need to consume about 100,000 of them in order to become ill. With this variant of E. coli, you need many, many fewer, typically between 1,000 to 10,000. So you need far fewer.
Are they more acid resistant?
Are they more acid? Not necessarily. You just need their virulence factors are just so much better at invading your gut and infecting your microvilli because of the pathogenicity island it has.
So what's the ultimate source? Cows of this E. coli?
Probably.
And the cows
fertilizer, manure.
And that's why you could also get it from a hamburger, right?
You can get it from hamburger, but there it's a direct result of inefficiencies in slaughtering and how many cows went into making that hamburger. If you go out to a fancy restaurant, how many cows were involved in the production of that hamburger? And the rule of thumb is you want one, but typically you sometimes can have between 10 and 100 different animals in the ground beef that's supplied to commercial restaurants.
So what you should take home from this TWIM is you should eat a lot less so you can live longer, and eat a lot less of these things that we've talked about.
That can make you sick.
Yes. Eating is a risk factor. For both.
Well, it's one of the hallmarks of the 20th century. We've really cleaned up our food supply. And while diarrheal diseases are still the number one killer on planet Earth, we've actually improved a lot in the 20th and 21st century.
Yep. All right. Cliff writes, Dear all, I'm a neuroscientist with an inventor's interest in both biology and technology, so I listen with delight to both your podcasts, TWIV and TWIM, and to your predecessors' podcasts, TWIT and TWIG. So TWIT is This Week in Tech, and TWIG is This Week in Google. And, of course, they are both produced by Leo Laporte, who was the inspiration for me to start doing a This Week in series. So thank you for that. And finally, Justin sends us an article published in Science. By wrapping itself in antibodies, this bacterium may become a stable, beneficial part of the gut. This is a cool summary of a story where they find that gut bacteria, I'm looking for what's the bacteria? Bacteroides fragilis. Its ability to stay in your intestine requires IgA, which coats the bacterium. It causes
IgA is our most produced immunoglobulin. It's like 40% of our immunoglobulins produced on a daily basis.
So the IgA coats the microbes and allows them to clump together along the gut lining and penetrate the mucus layer, allows them to stay right next to the cells. And without IgA, the microbes fail to colonize the gut. Very interesting. That is microbiology. It's an amazing field. Thank you, Justin. All right, that is TWIM 176. You can find it at any good podcast app on your phone or tablet. Just search for TWIM and subscribe so you get it every week. If you like what we do, think about contributing. Just $1 a month will really help us with our expenses. You can find out more at microbe.tv/contribute. And we love to get your questions and comments. Send them to TWIM at microbe.tv. Elio Schechter can be found at Small Things Considered. Thanks, Elio.
My pleasure. Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank ASM for their support of TWIM, Ray Ortega for his technical help, and Ronald Jenkins for the music you hear on TWIM. Thanks for listening, everyone. See you next time on This Week in Microbiology.